Hi guys, I'm Alethea Chekos, and this is my Animal Genetics Extra Credit video. My topic is meiosis. Meiosis creates gametes, which in turn is the foundation for genetic diversity. Let's clarify something. Mitosis, on your left, you will see that it only creates 2 daughter cells, whereas meiosis, after it duplicates, it creates 4 daughter cells. Prior to meiosis, a step called interphase happens, where G1, S, and G2 occur just like mitosis. The next phase is prophase. This is where leptema, zygonema, pachynemia, diplonema, and diakinesis all occur in small stages. An easy way I like to remember it is Led Zeppelin probably did drugs. To start off with, we have leptonemia. This is where chromosomes become visible, sister chromatids attach together through the centromere. Next, we have zygonemia and pachynemia. Zygonemia happens first, and this is where chromatids synapse. Basically, they just lay over each other in order for pachynemia to happen next. And that is where chiasmatas actually go ahead and switch, and this is where we get recombinant chromosomes. The last steps are diplonemia and diakinesis. Diplonemia happens first, and this is where chromosomes begin to move apart and the chiasmatas become more visible. After diplonemia, the rest of meiosis happens very, very quickly. The centrosomes help pull apart the chromosomes, and in diakinesis, the chiasmatas are terminalized. That means that the chromatids, they move to the end. This is one of the easiest times to count all the chromosomes in the cell. The next step of meiosis is metaphase, metaphase 1 to be exact. This is where the nuclear membrane is broken down, and then the chromosomes, they align up on the middle of the cell. The middle of the cell is called the metaplate. And then the centrosomes continue to form, and they create spindle apparatus, which then connects to the centromere of the chromosomes. And the next phase is anaphase 1. This is where the chromosomes actually separate. The chromosome pairs migrate towards opposite ends of the cell. The maternal and paternal homologous chromosomes actually go to different cells at random. The last step is telophase, and this is where the chromosomes have finally completed their migration, and they've begun to grow and create new nuclear membranes. At this point in time, there's 2 daughter cells, and they have been reduced to haploid number. If meiosis 1 are completed, meiosis 2, prophase, begins. It only begins in human females when the egg is fertilized. But in prophase, meiosis 2, the chromosomes condense, nuclear membranes break down, new centrosomes with their spindle apparatus begin to form. Just like before, we have metaphase next. And this is where the centromeres will divide and sister chromatids will separate by being lined up on the metaphase plate. During anaphase is when the chromosomes are actually pulled apart to opposite ends of the cell. This is by the centrosomes attaching to it through the spindle apparatus. Last step is telophase, and telophase is where a new nuclear membrane forms around the separated chromosomes, creating 4 completely individual randomized daughter cells. In telophase, we also have a process called cytokinesis that takes place. And that's all there is to know about meiosis. Thanks for watching.